This podcast is brought to you by Alice, Alistair, Eleanor, Luke, Nina and Veronica. Today we are going to discuss the range of animal models used in type 1 diabetes research. Animal models have been used not only to aid in treatment discovery, but also in the understanding of the pathophysiology of the disease. Researchers have been able to induce type 1 diabetes chemically and genetically in the aim of finding solutions to this intricate condition. Diabetes affects 9.2% of women and 9.8% of men worldwide, and the prevalence of the disease is increasing. The disease is characterized by an autoimmune-mediated destruction of beta cells in the islets of Langerhans in the pancreas. Through major histocompatibility complexes, the body comes to recognize these cells as foreign and consequently destroys them in a manner of ways. Although the disease is not genetically inherited, it is believed that individuals can have a genetic susceptibility. This, accompanied with an environmental stressor, can lead to disease onset. Symptoms of type 1 diabetes mellitus are as follows. Hyperglycemia. This is a high concentration of sugar in the blood and is a consequence of the lack of insulin production in the pancreas. Frequent urination also occurs. This is to try and reduce the excess blood glucose, also due to the lack of insulin production. In the long term, retinopathy, neuropathy and microvascular problems can also occur due to poor glycemic control. Currently, the only effective treatments are regular subcutaneous insulin injections.
Animal models have been used extensively in diabetes research. Early studies used dogs to confirm the central role of the pancreas in glucose homeostasis, resulting in the discovery and purification of insulin. Today, most experiments are carried out on rodents, although some studies are still performed on larger animals. Several toxins, including streptozotocin and aloxan, induce hyperglycemia in rats and mice. Selective inbreeding has produced several strains of animal that are considered reasonable models of type 1 diabetes, type 2 diabetes, and related phenotypes such as obesity and insulin resistance. Additional from their use in studying the pathogenesis of the disease and its complications, all new treatments for diabetes, including islet cell transplantation and preventative strategies, are initially investigated in animals. In recent years, molecular biological techniques have produced a large number of new animal models for the study of the disease, including knock-in, generalized knockout, and tissue-specific knockout mice. Animal experimentation has a long history in the field of diabetes research. In the 1880s, von Mering was working on the absorption of fat from the intestine when Minkowski suggested he remove the pancreas of a dog. The animal developed polyuria and polydipsia and was found to have diabetes mellitus. Many experiments on rabbits and dogs followed, although history has given a special place to Marjorie, one of the dogs used by Banting and Best in their seminal experiments on the isolation and purification of insulin in the 1920s. Marjorie was probably the most famous experimental animal in history, only to be superseded by Dolly the sheep in more recent years. Much of the research on islet cell transplantation has relied on the use of rodents previously rendered diabetic by experimental techniques. It is then possible to transplant islets and study the consequences. Islets may be transplanted with or without capsules designed to inhibit rejection, either subcutaneously under the renal capsule or seeded by the portal vein into the liver. Animals can then be given a variety of anti-rejection therapies. Even if human patients were to give informed consent for such studies, it is difficult to envisage how such a large number of experimental conditions could be replicated without using animal models. Nevertheless, it is important to remember that what works in a rat may not work in a human, and that the ultimate success of islet cell transplantation programs will be gouged by data from human studies. Furthermore, diabetic rodents are routinely used in the assessment of new pharmacological agents with a potential role for treating humans with the disease. Thus, all new orally active agents will be tested for efficacy and safety in rodent models. Sometimes this leads to potentially interesting and unexpected findings, other times less welcome results are obtained. At the outset, it is important to acknowledge that some people regard animal experimentation as an unjustified means of pursuing knowledge about human diabetes mellitus. There are those with the strongly held belief that it is morally indefensible for mankind to subject animals to experimental procedures that cause any degree of discomfort, as an animal is unable to provide informed consent and does not benefit directly nor indirectly from the studies. At the other extreme, a few medical scientists argue that they should be free to perform whatever experiments they consider appropriate with little or no external interference. In the United Kingdom, mainly as a result of public concern regarding vivisection on unanesthetized animals, a series of Acts of Parliament have been passed to control the use of animal experimentation. However, these control measures are different all around the world. In the United Kingdom, animal research is currently controlled by the Animals Scientific Procedures Act 1986. This defines protected animals as any living vertebrate except man and the common octopus. Any experimental or other scientific procedures applied to a protected animal which may have the effect of causing that animal pain, distress, suffering, or lasting harm are termed regulated procedures and are all illegal unless covered by appropriate licenses. Thus, in the UK, a researcher wishing to embark on diabetes research involving animals will have to be working in a designated establishment and apply for both project and personal licenses. Applications are reviewed by a local animal research ethics committee prior to submission to the Home Office, and a timescale of 6 to 12 months for full authorisation is not unusual. Recently, a high-profile group of UK academics have voiced concerns that they are increasingly unable to compete with research groups working in countries with far less stringent controls on animal experimentation. Even if a certain experiment is legal to perform in the United Kingdom, the administrative arrangements and costs may mean that the study can be performed more quickly and cheaply in another country. Furthermore, some diabetes charities that rely heavily on public donations may come under pressure not to fund animal research. For example, some donors may specifically seek reassurances that their money will not be used in animal research. Furthermore, animal research in the United Kingdom is governed by the 3 Rs: reduction, refinement, and replacement. Strenuous efforts should be made to reduce the number of animals required for any particular study. Experimental design should be refined so that it causes the least discomfort to the animals, and ultimately, animal models should be replaced altogether. Care should always be taken when comparing results from apparently similar experiments performed in different institutions and when extrapolating results from animal experiments to the human situation. Despite all these obstacles, significant advances in the treatment and understanding of diabetes have animal models to thank. These advances in knowledge have risen rapidly in recent years, especially with the advent of transgenics. It is likely that animal models will play an important role in the eventual cure of human diabetes mellitus, and it is these models that we will discuss in this podcast.
Spontaneous autoimmune animal models include nut mice and BB rats. Non-obese diabetic mouse was developed in 1974 in Japan. They are currently the most commonly used autoimmune model. This model is mainly used to understand the genetics of type 1 diabetes. Nut mice develop insulitis around 3 to 4 weeks. This is known as the pre-diabetic stage, where pancreatic islet cells are predominantly infiltrated by CD4 positive and CD8 positive T cells. insulitis causes the autoimmune beta cell destruction as seen in the human disease. However, onset of overt diabetes is not until 90% of pancreas insulin is lost at around 10 to 14 weeks. Diabetes is more prevalent in female colonies than male colonies, with 60 to 90% incidence and 10 to 30% incidence respectively. An advantage of the non-mouse animal model is that it displays many similar aspects to the human disease. For example, structural similarities of the MHC class II molecule are shared between non-mice and humans. Another similarity is the IGRP peptides. IGRP peptides were first identified to be targeted by isolate reactive CD8 positive T cells in non-mice. However, subsequent studies have shown that it was also targeted by CD8 positive T cells in type 1 diabetic patients. These parallels between non-mice and humans are extremely useful in understanding the genetic pathogenesis, the mechanism and pathway behind type 1 diabetes. Non-mice are also potentially suitable for testing therapies in which modulation of autoimmune responses is targeted. There are also limitations associated with the non-mouse animal model. A limitation is that many drugs that are effective in non-mice were found to be ineffective in humans. There are also difficulties in translating doses from non-mice to humans. Another limitation is when the pancreas of a non-mouse is removed for examination. There is a lack of biomarkers in the blood periphery in humans, therefore leading to difficulties in verifying the success of intervention. Non-mice also need to be kept in specific pathogen-free conditions. Due to gender differences, unpredictability of onset of diabetes, and requirement for SPF, these mice are expensive to maintain as a model for type 1 diabetes compared to chemical induction. Despite these limitations, non-mice is still used extensively as it does represent many aspects of the human disease and is a model that can help identify many genetics and signaling pathways that lead to type 1 diabetes.
A commonly used type of animal model for type 1 diabetes mellitus is the BB rat. There are two types, diabetes prone and diabetes resistant. Diabetes prone rats spontaneously develop autoimmune type 1 diabetes between 50 and 90 days of age. Diabetes resistant rats can be induced to develop type 1 diabetes under specific conditions. Both show the hypoglycemic phenotype in a predictable, short time frame, usually 14 to 21 days post-induction. In the diabetes-prone type of BB rat, pancreatic beta cells are selectively destroyed by immune cells, reflecting the human type 1 diabetes phenotype. T-cell lymphopenia due to mutation in the GIMAP5 gene contributes to selective beta cell destruction. Human type 1 diabetes has a closer phenotype to diabetes-prone rats than non-obese diabetic mice due to immune infiltration of beta cells, insulitis. In non-obese diabetic mice, peri-insulitis occurs. This makes diabetes-prone rats a better model of human type 1 diabetes than non-obese diabetic mice. There are advantages and disadvantages to the use of BB rats. An advantage is that they have a similar etiology to human type 1 diabetes and therefore have a strong face and predictive validity. Diabetes-resistant BB rats can have induced hyperglycemia, therefore allows determination of potential diabetes-inducible factors. A key disadvantage, however, is that BB rats are expensive to acquire.
In chemically induced models of type 1 diabetes, a high percentage of the endogenous beta cells are destroyed, and thus there is little endogenous insulin production, leading to hyperglycemia and weight loss. Chemically induced diabetes not only provides a simple and relatively cheap model of diabetes in rodents, but can also be used in higher animals. Diabetes is usually induced around 5 to 7 days prior to the start of the experiment to ensure stable hyperglycemia. Two main compounds are used to induce diabetes, streptozotocin or alloxan. Due to their similarity in structure to glucose, glucose can compete with alloxan and streptozotocin, and thus fasting animals tend to be more susceptible. Both alloxan and streptozotocin are relatively unstable, and the solution should ideally be made immediately prior to injection. Streptozotocin, due to its alkylating properties, causes alkylation and thus fragmentation of DNA. It modifies biological macromolecules and finally destroys beta cells, causing insulin-dependent-like diabetes. Intravenous injection of 60 mg per kilogram dose of streptozotocin in adult Wistar rats causes swelling of the pancreas followed by degradation of Langerhans island cells and induces experimental diabetes mellitus in the 2 to 4 days following. After 3 days of generation of beta cells, diabetes was induced in all animals. The NAD in the pancreatic islet beta cells caused histopathological effects in beta cells which led to diabetes. It should be noted, however, that it has been suggested that regeneration of pancreatic islet cells can occur after streptozotocin treatment, and thus sufficient control should be in place to determine that any improvement in glycemia is not due to spontaneous regeneration of endogenous beta cells. The procedure goes as follows. Male Wistar rats, typically in the weight range of 150 to 200 grams, were used, and 3 groups, a test, standard, and control, were all set up. The typical dosage administered was around 60 milligrams, and it was introduced via intravenous injection or intraperitoneal injection. Mice were used around 175 to 200 milligrams per kilogram a day. Dogs, 150 milligrams per kilogram for 3 days. Evaluation of the method goes as follows. Blood glucose level is estimated, and the results of which are compared to the standard and the control groups. Advantages of this chemically induced model include high selectivity towards beta cells, a low mortality rate, and longer diabetes induction. Disadvantages also include rabbits and guinea pigs are resistant to the diabetic effects. Streptozotocin has recently been shown to cause lymphopenia and a relative increase in T regulatory cells. One disadvantage with chemically induced diabetes is that chemicals can be toxic to other organs of the body. It should also be noted that change in P45 isosomes in the liver, kidney, lungs, intestines, testes, and brain have been reported after administration of streptozotocin or alloxan. Alloxan is another method of chemically induced diabetes, which produces a triphasic response. Phase 1. Early hyperglycemia of short duration, which lasts between 1 to 4 hours due to a sudden decrease or termination of insulin release and direct glucogenolytic effects on the liver. B. Phase 2 hyperglycemia phase, which lasts up to 48 hours and often results in convulsions. Phase 3, which is chronic diabetic phase, is a consequence of insulin lacking histologically only a few beta cells, if any are detectable in animals with fully developed alloxan diabetes. The diabetic effect of alloxan is mainly attributed to rapid uptake of beta cells and the formation of free radicals, which beta cells have poor defense mechanism to. A redox cycle for the generation of superoxide radicals that undergo dismutation to form hydrogen peroxide and thereafter highly reactive hydroxyl radicals that cause fragmentation of beta cell DNA. Advantages of this method is that they're taken up by the liver, but has better protection to reactive oxygen species, therefore it is less susceptible to damage. Limitations of this method include high mortality, ketosis, the diabetes induced is reversible, it cannot be done in guinea pigs, also alloxan has a narrow diabetogenic dose, and even light overdosing can generally cause toxicity, especially to the liver.
Mouse models of type 1 diabetes are useful for studying the pathogenesis and treatment of long-term complications associated with the disorder. Until the use of the Akita mouse model in the treatment of type 1 diabetes, no mouse model had been able to display the range of pathological characteristics of the disorder. The Akita mouse has been used as a genetically induced model of type 1 diabetes, in which symptoms of hyperglycemia and polyalbuminuria are observed. A genetic mutation in the insulin 2 gene was found to spontaneously occur in a colony of mice in Japan. This mutation resulted in a substitution of amino acids in the chain of mature insulin. Due to this, insulin in these mice was unable to fold correctly, resulting in endoplasmic reticulum stress. Mice heterozygous for this mutation were found to develop type 1 diabetes symptoms such as hyperglycemia at around 3 to 4 weeks of age. This mutation has proved extremely useful in diabetes research. Researchers have been able to induce insulin-dependent diabetes in these mice, which the immune functionality is extremely similar to that in humans. This is important as these mice have been used to test new formulations of insulin before being distributed to patients. Also, as the symptoms displayed are similar to that in humans, curative therapies such as beta cell replacement can also be trialled in these models. As the insulin-sensitive diabetes genetically induced in these mice is due to endoplasmic reticulum stress and not an inherent beta cell autoimmunity, the Akita mice was successfully used by Matthews in 2002 as a test for beta cell replacement protocols in vivo. These such studies highlight the advantages of this model over chemical models such as an STZ therapy. STZ therapy causes diabetes in animals secondary to pancreatic beta cell failure, as previously mentioned. However, it may be toxic to a variety of other tissues, which complicates the interpretation of results in studies regarding beta cell replacement. The human type 1 diabetes physiological environment is almost mirrored in the Akita mouse, and therefore these animals have been vital in the drug discovery process. Animal models of diabetes have provided invaluable insight into the pathogenesis of the human disease. Patients have directly benefited from the use of animals in the discovery of insulin and the assessment of other treatments. Animal research in the United Kingdom is governed by the 3 Rs: reduction, refinement, and replacement. Efforts should continuously be made to reduce the number of animals required in any particular study. And ultimately, with advances in technology, animals will hopefully be replaced altogether. Despite this, significant advances in knowledge have arisen in recent years, especially with the introduction of genetically modified animals. Animal models will play an important role in the eventual cure of human diabetes mellitus.